Hi, everyone, and welcome to our first ever Dandy Walker Alliance webinar. I am so excited to be joined today by Dr. Bill Loudon, the VP of Neuroscience at Bionaut Labs. Bill and his team at Bionaut Labs have developed a new remote-controlled microscale robot called a Bionaut, which can treat life-threatening central nervous system conditions by delivering therapies to regions of the brain that are otherwise difficult or impossible to access with current interventions. This Bionaut device can treat obstructive hydrocephalus associated with Dandy Walker in a less invasive way than the current standard of care with shunts and ETVs. But Bill is the expert here, so I'll let him explain it in more detail. Welcome, Bill, and thank you so much for taking the time to chat today.
Oh, thank you for the opportunity. We really appreciate it.
Yeah, thank you. I'd like to jump right in and learn a little bit about your background in neuroscience and how did you get involved with Bionaut Labs from there?
Sure, it's sort of a unusual story. I had kind of an unusual background in this. I am an adult and pediatric neurosurgeon. I've been in practice now for about 22 years, so not quite new at it. I'm still very active at it. But part of my training was a little more academically inclined. I was in graduate school. I finished a PhD in tumor biology at MD Anderson Cancer Institute in Houston. I was able to do an internship at the National Institutes of Health in Bethesda, Maryland, in the experimental neurology division, where I was working on adoptive immunotherapy protocols for, again, brain tumors. And over the course of time, I've had the opportunity to work with some really interesting companies like Medtronics and bringing some of their products to the clinic. So again, a little bit more of an unusual background. But the story as I remember it was way back in my residency, I was tasked with the job of helping a startup company that was working with magnetic systems to try and help them first with a catheter system that would navigate in blood vessels, the ability for the magnet to bend the catheter to be able to reach vessels that you couldn't coerce them to go into manually. And then the next step of that was to be an untethered device that you could use for surgical options. And we got going with that, but the company did not survive and kind of went away. And 25 years later, I was approached by a recruiter looking for someone with my skill set to work with this new company called Bionaut, which I'd never heard of before. And to just think it was interesting, it was sort of fate that the exact things that they're working on were something that I had spent a lot of my time thinking about and working through the problems so many years before. So I felt like it was just a continuation of my my previous experience.
Mm-hmm. It really does come full circle.
It really does.
And it certainly sounds like you've got quite a great background for approaching this, both from your clinical work and your previous work with the other startups. So Bionaut's lucky to have you, and we're lucky to be working with you.
Thank you.
So could you tell me a little bit about the Bionaut technology? What is it, and how does it work?
Right. So it's addressing some great unmet needs in medicine. And I'm going to be clear from the beginning, although we're focusing on the nervous system, the initial envisionment of this technology was actually looking at other organ systems, which still may prove to be interesting down the road: liver, eye, lungs, things like that. But again, the extent of the unmet need in the central nervous system is so great, and the challenge is so high that the decision was really made to focus on this. Again, much to my taste, I'm really appreciative that we did go this route. So here's the deal. The brain and the spinal cord are locked within protective walls. The dura that surrounds it, the skull, the spine. All these things were designed to keep outside things out. And The problem with that is that surgeries to get to the brain tend to be relatively aggressive and fairly in depth, and carry a lot of risks associated with them and a lot of recovery time for that purpose. And then drug delivery on the other side, we have blood-brain barriers and other very significant problems to try to get drugs to where we want it to go. So both on the surgical side, it's hard to get into the brain, and on the drug side, it's hard to get into the tissues. So the Bionaut has specific advantages that circumvent these problems. And we can go into those in our discussion, but from a surgical perspective, we have been moving towards what we call minimally invasive surgery. And that's just what it sounds like. Instead of these giant incisions we used to make, taking the bone off and doing these very, very aggressive surgeries, we're now getting to the point where we make little incisions and are able to accomplish the same ends through these much less invasive, much less painful, much less dangerous procedures. And I think that is an ongoing process. And in my mind, Bionaut represents truly a whole new level of this minimally invasive concept.
Mhm. And how does it specifically treat the obstructive hydrocephalus that's
Sure.
associated with Dandy Walker?
Yeah, it's a great question. Let's go back in time to the late 60s and early 70s. Hydrocephalus has been and remains a real medical challenge throughout the ages, right? Hydrocephalus has been expressed or described way back in time and history. And as you can imagine, there have been some very unusual, creative ways to try and treat hydrocephalus, but not until the 60s and 70s did we actually come up with ways to divert fluid from the brain, for example, to the heart or to the abdomen, to the development of shunts. That was relatively late in the realm of medicine. And before that, we were really required to do open surgeries. And so the obstructive hydrocephalus associated with Dandy Walker is a unique expression of the abnormal anatomy of Dandy Walker. There is literally a blockage for the outflow of CSF at the last point in the brain. So CSF is made inside the brain, circulates through the chambers, the ventricles of the brain, ultimately to the fourth ventricle right before it exits and goes around the brain and is absorbed back into the bloodstream. The cycle repeats over and over. And Dandy Walker, in some cases, about 80%, there is an actual membrane, it's a wall associated with the cyst that blocks the fluid at this last point from getting out. So the fluid accumulates under pressure and causes the symptoms of hydrocephalus. So in the days prior to the development of shunts, the surgeon was required to make the decision of whether or not it was worthwhile to do the large surgery, opening up the back of the head, removing the bone, opening the dura, going under the back part of the cerebellum, and physically cutting a hole through the window, as a window into the cyst to try and reestablish the flow. Sometimes it worked, sometimes it didn't work, but you can imagine there was a significant amount of morbidity associated with that aggressive surgery. So sure enough, when the shunts actually hit the medical arena, the ability to do a much less aggressive surgery became very attractive. So we're now putting tubes in that are upstream of the block, and that allows the fluid to be drained that way. So in a sense, it's a hack, right? It's not fixing the problem. It's sort of redirecting the flow away from there. And
Exactly.
if shunts were foolproof, it would be wonderful, right? We'd have no reason to look elsewhere. But I think anyone listening to this presentation who is aware of the issues associated with shunts are probably very aware of the concerns that, shunts, as beautiful as they are, as wonderful and life-saving as they are, they break, they become plugged. They stop working for a number of reasons, or they don't work in an appropriate manner. They drain too much, they drain too little, and even worse, they can cause bleeding, they can cause injury to the brain, and they can become infected and. So, patients with complex hydrocephalus, as are many of the Dandy Walker patients, have a long history of shunt-related problems and shunt-related surgeries. So what we're looking for here is to go back to the concept of creating an anatomical repair not a hack, not an indirect, but an actual anatomic repair, by now being able to do a very minimally invasive procedure to go right to the base of the cyst, through the skin, into that space, just under the edge of the bone, and open up that cyst wall to let the fluid now drain out in a normal fashion. That's the overall plan with using the Bionaut technique, this little mini robot to be able to travel to the cyst wall, fenestrate or cut open the wall, and allow the fluid to drain normally again. So we hope that it can serve as an adjunct or maybe even an alternative to shunting procedures.
That's fantastic. And obviously, there's a ton of potential uses for this Bionaut technology, and I know that you're also looking at pediatric brain tumors as one of the other
Right.
uses for this. But what made you focus on obstructive hydrocephalus as one of the initial conditions that you wanted to work on?
So again, it's a very long discussion in and of itself, but let's start by simply acknowledging that the technology we're talking about, a little tiny micro robot that's introduced into the cerebrospinal fluid or the brain and then travels based on controller systems, that's a very novel concept, right? And in the world of the FDA, because it's novel, it has no precedence, it automatically falls into a high risk category, but as it should, right? It's a completely new and novel system. So to facilitate FDA approval, we are looking to separate the device itself from other functions, such as the ability of it to carry payloads, to carry drugs with it to targets in the brain. So the simplest pathway to this, and again, this has hours and hours are spent with consultants from the FDA and with the FDA themselves on arriving at this plan. The first step here is to use it as simply a device to prove that it's effective and safe. And that, again, is the Dandy Walker. That's why we've arrived at that point. But I want you, again, to think of Dandy Walker as one small subset of obstructive hydrocephalus overall. Does that make sense? So it is the stepping stone that allows us then to move into much more complicated forms of hydrocephalus as a device and other surgical applications. When all of that is proven safe and effective, then we add the other component, the ability for it to carry therapeutic agents to hard to get to places in the brain as the next step. So device drug combination versus right now starting with the device alone.
That makes sense. And it sounds like this form of hydrocephalus associated with Dandy Walker is a really good way to start using the Bionaut product to change the lives of people around the world.
It really represents an ideal first step for this. And again, I think the concept of reestablishing normal, CSF flow patterns is very appealing to us, especially for patients who have had numerous problems with their shunts.
So, kind of going off of that last comment, I think a lot of people in this community are familiar with the shunt treatments or even the EVT treatments. Will the Bionaut procedure be safer than a shunt or an EVT? And what will the hospital stay and the recovery process afterward look like?
So let's be clear on this, right? We are by no means into the clinical arena yet. And, please, I hope nobody's taking this as an attempt to recruit patients for a study yet. We are excuse me, we are at the very basic stages right now of addressing all of these points. And just to be clear, the pathway for us is starting with in vitro models of this, that we work this all out to every degree that we can in model systems before we ever contemplate moving into the clinical arena. So we're still quite some ways away from that. Our hopes are this will prove comparable to the efficacy of shunts, and we hope that we'll be able to show the safety, the efficacy, and the durability. Those are absolutely our goals. But the first goal obviously is safety. We have to show that this is unerringly safe. And then the next step is that we actually can make an opening in the cyst wall. And so those are our first two real goals. And then, everything else beyond that is conjecture at this point. But you can imagine that, a small needle inserted at the back of the neck to introduce the Bionaut has a much reduced morbidity to it than does an open surgery. So we would hope to think that we would have reduced hospital stays and a lot of other benefits from, again, moving down this path of minimally invasive surgery.
Right. I think the minimally invasive is what will likely be appealing to a lot of people in our community and around the world, for people both being treated for hydrocephalus and I'm sure for some of the other conditions that you're focusing in on as well. Do you have any idea of how you plan to measure whether or not the procedure works, like CT scans after the surgery, MRIs, anything along
Right.
those lines?
So there are multiple levels to that question. The one that we have to focus on first for the FDA is we have to, at the time of surgery, prove that we've effectively made this opening, this fenestration. And there are two ways that we are looking to do this that build on each other. Again, one of the simplest ones to understand is we also have the ability to use a little endoscope, a viewing telescope of sorts, to be able to actually look at the product of the work, right? To be able to look at and see that, yes, we've generated a hole that communicates.
Right.
Other ways of adding to that are the ability to deliver a small amount of contrast agent into the fluid, and after the procedure, see it get into the cyst where it couldn't get into it before, right? Long-term proof of efficacy will obviously fall into the realm of imaging studies, but most importantly, how's the patient doing, right? Are their symptoms relieved? That's obviously what we're really shooting for here, is to improve the quality of life of the patient with the Dandy Walker cyst.
And in the scenario where this Bionaut procedure is done in clinics and is seen as one of the standard of cares, let's say the fenestration closes, can the procedure be done again, or is it kind of like a one-time?
Good question. Yeah, so let's use endoscopic third ventriculostomies as an example. Many patients now are undergoing this procedure to help with the hydrocephalus with Dandy Walker. And a small percentage of them have to undergo this repeat time because the opening, the fenestration will close. And this is completely analogous to what we see for the Bionaut approach to the Dandy Walker cyst fenestration. So yeah, we totally envision the ability to go back in and repeat the procedure if necessary.
That's great. And obviously, you have to ask, is there any potential for damage to the brain by piercing the cyst to drain the excess CSF?
Sure. So again, we can start from kind of where we are standard of care right now. With insertion of shunts or revisions of shunts, we have all the risks. We have risk of infection, leak, of injury to the brain, bleeds, seizure, stroke, all of those things from the manipulation with the mechanical parts of the shunt. Same thing's true with endoscopic third ventriculostomy. We're still having to drill a hole in the skull, pass through the brain, move into the CSF space, make a hole in the cyst wall, try not to damage things around it. So what we're looking at is, by being able to safely direct the Bionaut without having to go through the skull, without having to go through brain tissue, to not have to touch any of those vital structures, simply starting in the cerebral spinal fluid, moving the very short distance to the cyst wall, which cyst has no neurologic function at all. To be able to open that up and come back out safely, we hope that it will very favorably express itself as a very safe procedure. Again, that has to be unequivocally, that has to be the number one most important aspect of this new technology.
Well, I love hearing how much of an emphasis that you all are putting on the safety of the
Mhm.
patients. And I think for a lot of people in our community who have so many questions, both with the current standard of care and any new procedures, I think that'll likely help put them at ease. But
As it should, as it should.
mhm. So obviously you're in the very early phases of this product, but what do you see the next steps being for the development and potential clinical trials down the line?
Sure. Yeah. So it's again, this is a very well orchestrated plan that is true to all new devices. There's a very specific pathway with the FDA, partnership with the FDA, to bring us through a series of studies, both in vitro and then preclinical studies, and then ultimately escalating into actual clinical trials. But we have a number of hurdles still to achieve to the FDA's satisfaction before we
Hmm.
would ever be considered the IDE, essentially their permission for us to proceed with clinical trials. Once we start the clinical trials, then of course the absolute scrutiny on how things go, how we do, is much more intense even than previously. But it's because we have no precedents and we are therefore a high risk device. We have the highest bar to reach every step of the way. And
Hmm.
so by the time we get to the opportunity to start clinical trials in patients, we're going to feel pretty good about our platform.
Hmm. That's that's great, and I'm sure there's numerous steps between here and there, but
Mhm.
um, I know that we're very optimistic in the work that you all are doing, and hopefully, we're in a place down the line where this becomes standard of care for many patients.
Hope so.
Um, and do you have any projections? You know, what the path would look like going from clinical trials to being used as a standard of care as an alternative to a shunt or the other
Right.
procedures?
So there, it's sort of a moving target, as you can imagine,
Mhm.
right? At first, we have the goal of treating a few patients and get safety. And at least the primary objective is to open the cyst wall at that time, to prove that it's
Mhm.
open at that time. Once we have successfully, safely done this in a few patients, then the overall equation of the clinical trial will hopefully change for us with the FDA supervising this. And it can then expand much more rapidly to a broader definition of obstructive hydrocephalus. So then
Mhm.
we can move beyond just the Dandy-Walker membrane. We can move into other cysts, for example, complex hydrocephalus in the ventricles above. And it will be, in my mind, at least in my hopes, a very rapidly expanding clinical trial at that time. To be clear on that, as you can imagine, the workload involved in this, we have been reaching out and are developing collaborators at the top pediatric neurosurgical institutions in the country. And they will be our partners in this as we move forward. And so we already have very significant interest and buy-in by these centers. These are very busy neurosurgeons who are taking time out of their schedules to visit us in our labs here, and they're hands-on working with us to develop the equipment. They're listening to the strategies and the protocols, and everyone is participating in this. This is perhaps the most rewarding thing for me so far, is the validation of Bionaut's perception of how this is going to be. We have these are very respected and very accomplished neurosurgeons in the country, the best of the best. And the overall excitement and interest in what we're doing has been universal. So
Mhm.
they all want to have this platform to be able to use it in their practices, and to have that type of momentum behind us, you can imagine how rapidly we could expand our applications. It's incredibly exciting. You can imagine how nervous I was when I first started presenting this to my colleagues. It's like they're going to throw tomatoes at me. This is so crazy. And we have comments, we have videos that I've recorded of my colleagues to share with some of our non-medical people here. And I love the comments. It's like, this is the craziest thing I've ever heard of. I can't believe we haven't thought of this before. And just the overall excitement and the acceptance of this, and you can just see people rethinking all their problems with this. And again, we could spend, hopefully maybe down the road, we can talk more about why this represents such a radical change in the sphere of minimally invasive surgery from what we've been restricted to in the past. It opens the doors to just whole new ways of thinking about things.
Mm-hmm. Well, I can tell from the excitement on your face talking about this that this really is a momentous step in the field of neuroscience and neurosurgery, and with the potential, like you said early on, to impact other organs and systems throughout the body. And I think it's really exciting what you all are working on, and we're just so excited to be a part of it at the Dandy Walker Alliance as well.
Great.
So as we start to wrap up here, I know that we've put out a lot of information to our constituency about how they can get involved with providing MRIs and imaging, but I'd love to hear from you more about how the Dandy Walker community can support the development of the Bionauts going forward.
Right, and this is I can't communicate enough how important this relationship is with the Dandy Walker Alliance. As family and patients with Dandy Walker understand, it's a very rare disease, and as a rare disease, it doesn't attract a lot of support and attention as would other. I mean, compare Dandy Walker to Alzheimer's, for example, right? Billions and billions of dollars going into Alzheimer's, and we just don't see that kind of support with Dandy Walker. One, of course, because it is such a rare disease. So the ability to have a motivated community that is willing to support us, to work with us, to partner with us, to move this forward is not just great, it's essential. We're not going to be able to do this without your support. And so this is a partnership in my mind. And I have spent a lot of effort making sure that my colleagues at Bionaut understand that to be able to ultimately have access to the patients, the Dandy Walker patients, is going to require the community to rally around us to do this. So the more that we can educate and involve, get feedback from the Dandy Walker community as we move forward, I think the better able we'll be to move forward into the clinical arena. The images, for example, because they're so rare, we're desperately trying to get as many of these images as possible. And essentially, the engineers are running it through our segmenting protocols to obtain measurements, and we'll try to overall generate parameters of what is Dandy Walker and how do our instruments fit into the parameters of Dandy Walker, right? You can't just stick something into a brain, especially not something as complicated as this. You have to really understand what it is. And you can't make this stuff up. It just is. You have to actually get the biology. You have to get the actual patient information to be able to really build this understanding of what Dandy Walker is.
Well, it's very exciting that individuals and families in our community have the opportunity to make such a real impact on the development of this technology that can be life-changing for them and for numerous other people in our community down the line. So I'm very excited to have this partnership with Bionaut Labs and very excited to be working with you. Before we wrap up here, is there anything else that the Dandy Walker community should know about your work, Bill, and the Bionaut technology?
No, I think that it's it's a really exciting moment at this point in time, the ability to really be on the verge of being able to offer something that's never been available before, to be able to bring a truly minimally invasive technology to bear that is going to be first tested and proven in the Dandy Walk arena, but ultimately then expand out from there. So the beginnings of this will all stem from the Dandy Walker population. And what we learn here is going to benefit many, many other patients out there with diverse issues, other CNS pathology. So again, this partnership has so much meaning and relevance to it that I hope that we can really share in this, in the progress as we move forward.
Mhm. Well, Bill, we are so thrilled to be working with Bionaut and so excited about all the work that you and the rest of your team there are doing, and look forward to growing this partnership and helping develop this product and bringing it forward to where it can really impact the lives of people all around the world with all sorts of different conditions. So
Right.
thank you so much for the work that you're doing.
I really appreciate the opportunity to discuss this with you and look forward to be able to have more forums like this in the future, to be able to share this and to address questions and concerns and learn from the community how to better reach out to them. Thank you.
Well, Bill, thank you again for taking the time to chat and inform our community about the exciting work that you all are doing at Bionaut Labs and the potential this has to change so many lives in our community and beyond around the world. And it's very exciting that people in our community have the chance to get involved and make an impact. So if you're tuning into this and you have obstructive hydrocephalus caused by Dandy Walker and you want to get involved, I would really encourage you to. Like Bill said, they're looking for MRIs and images that can help their engineering team train the bionauts to understand the cyst anatomy in the brain. So if you're interested in learning more about the technology, connecting with Bill, getting involved, sending in images, I recommend you reach out to clinical@bionautlabs.com and they'll get the whole process started for you. So thank you again to everyone in our community for tuning in to this. And thank you so much to Bill for taking the time to share the great work that he's doing at Bionaut Labs. Until next time, take care.